In our last session, so we are going to, sorry, someone is speaking. Okay. Hi again, everyone. So today we are going to start our last session. So what we are going to discuss today is de novo transcripts assembly. So de novo transcripts assembly is an approach that is being used for those kind of RNA-seq data that do not have any reference genome. For example, you have just developed a new variety or new cultivar of a plant species, then to see do RNA-seq analysis of that specific cultivar that you have just recently developed. Within a certain condition, you want to see okay, which gene has been expressed or which has not. So you extract RNA and then you do analysis using bioinformatic tools. But no, you don't have any reference genome. So you don't have as any reference genome. So how you gonna align your reads? So we use for this kind of problem to address this kind of problem, we use de novo transcript assembly. So sorry, someone is want to ask something.
No, sir, please proceed.
Okay, so the first step what we do, so we every time we are using we are getting RNA-seq data, so it should be in the form of FASTQ files, and if the FASTQ files, and maybe these can be compressed. So if the files are compressed, then you will see a dot GZ. So this indicates that the files are in FASTQ format, and but these are compressed. So you can uncompress these files, or you can proceed with the compressed files. So after that, similarly, we will do trimming and filtering. So we have Trimomatic or cut adopt to remove low quality reads and adapter sequences. So here is the simple command how we can use this Trimomatic for removing adapters and filtering low quality read. So next thing what we do, we need to remove ribosomal RNA. You know, when we are extracting RNA, so we get with total RNA, we also get some ribosomal RNA. So that ribosomal RNA is totally unwanted. So what we would need to do, we should remove that unwanted RNA. So removing unwanted RNA is helpful so that it can reduce our data size, and it can make it and it can improve its quality. So it's easy to proceed. So after that, you have removed unwanted ribosomal RNA. So next, what you do is a technique what we call it de novo assembly. So mostly the package, most famous package that is being used for de novo assembly is Trinity. So Trinity is a big package. So it also needs some associated packages. So before installation, after installation of Trinity, so we need to make sure that we also have Botai 2, RSEM, SAMon, and SAM tools. So we also need to add these packages in our Linux environment. So these packages will help Trinity to do a de novo assembly. So how we gonna start? So you can see there's a really simple command. So you will write Trinity, then you need to write how many CPU you are using. So you can see in this command you are using 8 CPUs. So this option is used when you're using cluster computer. So cluster computer is like it's a big computer that has tons of computational resources, CPUs and RAM, and then you can allocate how much you want to use on that cluster computer. And then you have to mention, okay, what type of data you are trying to assemble, and the type is sequence data, and then you need to put the file format, what type of format it is. So if it is a FASTQ file, you can put FQ, or either you can directly put here FASTQ, and then how much RAM you want to use, max memory you want to use, and then you have to mention, okay, this one is a forward sequence and this one is the reverse sequence, and then what output file name you want to keep. So here's a general example. You want to keep output name, file name, trinity output. Okay. The other method you can use, if you have multiple files, you can see there's a just one sample. And if you have multiple files, samples, then what you do, you create a text file and put the sample's name, and you can directly upload your sample file. So these are two methods to run this command. So when you after finishing this command, you will get your output in Trinity output directory. Directory means folder. So in Linux we call a folder a directory. Okay. Next, when we got the assembled sequence assembled Trinity data, so next we need to apply some quality parameter to see, okay, uh, what is the quality of transcript what we got. So one thing you keep in mind, this step is time consuming. Keeping in mind your computational resources and how much is the size of your input file. So when I was doing my own data analysis, de novo assembly, so the size of my data was around, I think 200, 250 to 300 gigabytes. And the RAM I was using, it was 1.5 TB RAM, and I was using around 32 CPUs. So using all these resources, it took around 2.5 days. So if you are using your own computer with limited resources, so you can accept it is going to take a while. So next, what we are going to do is apply some quality parameters. So we will run this script to see what is the quality of our transcript. So this text file will give you stats that will show, okay, how much transcript has been assembled. Okay. Next, what we are going to do, we are going to use other packages, Busco to see, okay, what how we can see the completeness of this data. So for checking completeness of data, we use package called Busco. So either you can directly download using this link. I have also pasted here link to read the guideline. Okay, and then you will run this Python script. So .py means it's a Python script because Python is already in your environment, so you don't need to install Python again. So you will run this command. It will be executed automatically. So next, what we will do, we will remove the redundant sequences. So whatever redundant sequences are in your Trinity assembled assembled sequence, you're gonna remove it. Okay. So what's next you are doing? So next you are what you are doing, you are what is called blasting your sequences. So you blast your sequence using a Ugene package. So it's called CD hit. So you use CD hit and blast, get the blast results. And next you measure, okay, what is the distribution of your sequence length? So it's a script for to see the distribution. So all these are after you get the Trinity assembled file, all these are to evaluate the quality. So next step, it is really important. It is annotation of script. So annotations means no, you have assembled your transcript. You don't know what are the genes on in those sequences. So for that purpose, you do annotation. Annotations mean to give name to your coding sequences. So it is a really long process. So what we use for annotation, so we use first Simon to quantify transcript abundance from the assembled transcriptome. So this one is the command, Simon command. And next step, you use trans decoder. So trans decoder is another package you use to identify candidate coding regions within the Trinity assembled transcript. And for annotation, you use trinotate. So trinotate, we use trinotate. For using trinotate, we need to generate other files. So what file we generate? First, we need to generate a trinotate SQLite database. So here's the command. Okay. So when we do blast, so after blast, you get these kind of files. You do blast x, blast p, and then blast with pfem and run signal IP and then THM, and you combine all results and get a single file. That's called blast result output. And then you need to load your results using trinotate. So these are the all file you combine the output of all these file into one single file that is called annotation report.xl. Then you will generate Excel file. So once you have generated Excel file, then you can do what differential expression. So this one is the one method for using doing transcriptome annotation. So there is another package called eggnog mapper. So this can be also used for uh, transcript assembly. So these are the all commands for eggnogger mapper. So you can directly download it and read its instructions on GitHub page or directly on its website. So Eggnogger can be installed by using a simple conda installation method. Okay. So first you need to download database and export it into your environment. And then you need to run this command. And then the other one. Okay. So there's a third method you can also use in interproscan. So interproscan can be used for transcript annotations. So these are the three methods you can use for transcript annotation. So these are the codes for doing transcript annotation using interproscan. And finally, you can use any one method, then you will get the Excel sheet or text file. Then you have to do a differential expression. So differential expression, you can use R D sig2 package in R. So I have also mentioned here the general transcript script of R package that you can use for doing a differential expression. Okay. So next, you can do visualizations with there are many visualization packages in R. You can use ggplot2 and build volcano plots to see your uh, upregulate to separate upregulated or downregulated data. Okay. So next step is you do a pathway enrichment analysis. So there's a two method you can do pathway enrichment analysis. One is you can use tool like GSEA or David, and there are other that you can use like Shiny Go. So Shiny Go. It is really easy to use. If you Google it, Shiny Go, so you can upload your transcriptomic data and you can get really beautiful visualizations. So this is how we can do de novo transcripts assembly. Is there any question so far?
Hello, sir. Assalamu alaikum.
Yes, yes, Rafia.
Sir, when yesterday you have told to download the FASTQ file of RNA set from the NCBI. So when I am downloading, they ask for the SRA toolkit.
Sorry, I asked you. There is a NCBI's file section that is called SRA. So using SRA, you can download already published or already done all RNA-seq data. So there's a two method you can use. One is you can directly download in your environment using SRA toolkit. And other is you can download it and then upload it. So
Okay,
yeah, you can install SRA toolkit in your Conda environment.
okay, sir. Sir,
Yeah, that
okay. Please share
yeah.
the
Sorry.
please share the command for the downloading the SRA toolkit in the
Okay, okay,
environment.
okay, okay. Let me share it. So prefetch command is in a SRA toolkit. I have just pasted in a chat. So if you just accession number type prefetch and type the accession number of any SRA file, then it will automatically download in your environment. Okay. Any questions? So no, we're gonna move forward to our next and
I have a question.
yes.
Uh, this number SRR309, it's a specific number for an RNA sequence in the database. Do we have
That's a good question. Let me share the screen.
to?
Okay. Can you see my screen?
Yes, yes.
Okay. So if you type in Google NCBI SRA, then you will be finally here on SRA window. So if you type Arabidopsis, I just type Arabidopsis, and then there are many uploaded RNA-seq data. So what I'm going to do, I'm going to click on anyone, and you can see there's an accession number. Okay. If you copy this accession number and type that command, prefetch, and then type the accession number of any sequence, you can see every data sequence has their accession number.
Okay, okay.
Okay. Then you can download it.
Okay, thank you.
No, yeah, no problem. So just keep in mind these data sets are already in the NCBI, and they have performed some experiments. Some have exposed Arabidopsis to heat, some has exposed to cold, and this data will be really
So then
specific.
the next class, we will continue about the homeostasis. I think we started about the homeostasis. Did we discuss homeostasis? No. No, we were talking about hormones. Yeah, temperature regulation. Yeah, we only talk.
Okay. Now we are moving to our last session, that which is doing variant call analysis. Okay. Initial steps are almost similar as uh, as we have already learned that first of all, either you have genomic sequence or transcriptomic sequence. You need to do quality analysis. So we are gonna use the same fast QC package to see the quality, and then use the Trimo Trimotech or cut adopt to remove low quality reads and adapter sequences. Then we need to do alignment. So we can use a star package for analyzing our genome sequence or transcriptome sequence to reference genome. So after alignment with the star, we're gonna do post alignment quality control check. So we can use SAM tools to index the BAM file and perform quality check. So here's a command to indexing your aligned BAM sequence using SAM tools. So next, what we're gonna do in step 5 is marking duplicates. So marking duplicates, we use the package called PyCard. So here's a command for marking duplicates, and you will get a text file at the end. Okay. So this step is specific if you have RNA-seq data. So this command will split reads into exonic regions and trim NCGAR's elements. So next, what we're gonna do, base quality score recalibration. So the package we use is a GATK, and then we applied the recalibration to the BAM files. And in step 8, we're gonna find the variant calling. So we use this package GATK haplotype caller to call variant from the recalibrated BAM file. So this one is the command. And then after you got the variants, then you do filtration of your variants. So here's the command for filtering the variants, and then next step is annotation. So the variants you annotate them. So for annotations, you use SNP EFF. So this is a big package. It contains almost all basic organisms annotations, including Arabidopsis, humans, mice, rice, cotton, many others. So you can do annotations of variant calls you got. And once you have got your VCF file, which is already annotated, then you can use IGV browser to visualize your variants. So this is the whole pipeline. You can find the variants in your data sets when you want to do single nucleotide polymorphism on large data sets, which is in the GBs. So I have wrote these pipelines in a really generic way. So when you are doing analysis, so based on your sample name, based on your file names, you need to modify everything. So it is a bit tough but when you do practice, you will learn step by step. So any questions so far? Okay. So I think this was the everything from our workshop. So mostly my focus was, okay, you need to first set up the environments and install the necessary packages. And then once you have pipeline, you have data, so you can easily play with it and you can do your analysis easily based on your, if you have plenty of computational resources, you can do it early. But if you have limited computational analysis, just keep in mind, limited computational analysis resources doesn't mean that you cannot perform it. Even you have 1 TB hard drive and 16 GB RAM, you can easily perform it. But it will take some time. Even sometime it's gonna take 2 3 weeks to perform a single task. Okay. When you start doing, if you have any specific question, so you can reach out to me. So I will share my email address in the group. So you can email me if you have any difficulty.
Hello, sir.
Okay.
Assalamualaikum.
Uh, yes,
Sir,
Rafia. So I can, yeah, I can see you are
yes,
downloading the data. So yeah,
yes,
it is
sir.
downloading.
It's download or it's downloading?
It is downloading. You can see. So it's a simple thing. If you are not able to type anything, it means command is running.
Okay, sir.
So it is downloading data.
Sir,
So
but it's
yeah, just keep in mind the data you are trying to download, it will be a big file. It will be around GBs.
okay.
Yeah.
Hello, can you hear me?
Yes, I can hear you.
Uh, I am facing a problem. I'm going to share my screen. Please help me out with this.
Okay, sure. You can share your screen.
Now I'm trying to refresh the accession number, but continuously you can see the statement which appears on my system. Connection busy while
So
validating within
so
network
so
system.
first you need to create an environment. You have created your mini Conda environment.
Yes, I have.
Okay. Can you please type the command?
For what?
Okay, let me type a command in the
Yeah.
chat. Yes, press enter. Don't write this. Just press enter.
Just in.
No, no, just remove en at the end. Okay. You have created many environment.
Yes.
Okay. Okay. Can you type conda activate en88?
Now press enter.
Yes. Okay. No, try to install SRA toolkit. I have just pasted command in chat.
नहीं ले रही हूँ प्लीज ये आवाजें ना आए
Okay. Okay. Now try to run the prefetch command.
and okay internet connection unstable है
Yeah, it's you have unstable internet connection.
okay
Yeah.
okay I will try it later on yeah
Yeah, sure. Sure. If you have any problem, you can reach out to me via email.
okay thank you very much
No problem. Okay, I think we are now going to end this session. So thank you so much, everyone, for being part of this workshop. I hope you have learned some basic regarding Linux, and I'm sure it will help you on later on. If anyone need any help, feel free to reach out to me. So I will be happy to help. So if you have any question, please ask. Otherwise, I'm gonna end the session. Okay, everyone. Have a good day. Bye.